If you'll get your second diagram that's got the number 2 and it says membrane proteins for ETC. These are the proteins that are in the gradient that the electrons pass down. Complex 1, CoQ, complex 2, complex 3, cytochrome C, complex 4, and lastly oxygen. Now, in a more advanced class, you'll have to learn the particular names of each of those proteins that are involved in ETC. But for our class, all you need to know is complex 1, complex 2, and oxygen. Now, complex 1, you'll remember, is where the high energy buses unload their hydrogen electrons. Complex 2 is where the low energy buses unload their hydrogen electrons. So put a star by those two. You have to know the names of those two compounds and what occurs there for the test. And then the very last compound that receives is oxidized by the hydrogen electrons and also receives these protons that are going to come back through this gate here, is oxygen. So the last hydrogen acceptor is oxygen. Oxygen joins with the hydrogen electrons and protons and forms metabolic water. Now, immediately an enzyme chops this water into hydrogen protons and a hydroxyl ion, and hydroxyl ions carry a negative charge. So on the inner compartment here, we have all of these hydroxyl ions. Now, as the electrons go down the gradient, all of these chemical reactions that are occurring release heat. That's a form of energy. And this energy is used as a proton pump, and it pumps the hydrogen protons that were on our buses from the inner compartment to the outer compartment. Now those proton pumps are at complex 1, at complex 3, and at complex 4. So complex 1, 3, and 4 are hydrogen proton pumps. Now what happens is that as all of these hydrogen protons are pumped out, an electrical charge is formed here in the outer compartment, and that electrical charge is positive. Now on the inner chamber, or inner compartment, we've got an overall negative charge. So this creates kind of like a little battery here in the mitochondrion. Now, these hydrogen protons cannot pass through the crystal except at one point, and that's right here in this little gateway that's called ATP synthase. And we'll see that on the next diagram. So, the source of hydroxyl is this metabolic water, and the hydrogen protons are pumped out in 3 places along this gradient, at complex 1, at complex 3, and at complex 4. Now get your handout number 3, which is hydrogen electron and hydrogen proton movements in ETC. Okay, here's our energy gradient. The buses, this is a high energy bus, dump their electrons to complex 1, and then it goes down the gradient. All of the buses that are high energy unload their hydrogen electrons at complex 1. Now, if it's a low energy bus like FAD or the NAD H plus H's that were in glycolysis, they unload their hydrogen electrons at complex 2, and then they go down the gradient from high to low. So electrons, hydrogen electrons, go down the gradient. The protons are pumped from the inner compartment rather, to the outer compartment, and they're pumped out at complex 1, at complex 3, and at complex 4. So, hydrogen electrons go down the gradient. Hydrogen protons are pumped out at the C1, C3, and C4. Now, remember, the protons cannot pass through the Christie except at one point right here at the end. So what we end up is a high concentration of positive charges in the outer compartment and negative charges on the inner compartment. So remember, electrons go down the gradient, protons are pumped out. Now find your next outline, which says chemiosmosis, ATP formation. All right. So, what we've got are all of these hydrogen electrons that are out here. And if you're a high energy bus and unload your hydrogen electrons here at the top of the gradient and they go all the way down, a high energy bus can produce 3 ATPs. If you're a low energy bus though, you skip this first step. The hydrogen electrons come in here and go down, and low energy buses can only make 2 ATPs. So, remember, our Christie is impermeable to hydrogen protons except right here. Now, this protein is called ATP synthase. ATP synthase is where the hydrogen protons re-enter the inner compartment. The hydrogen protons come in 2 at a time. They pass through the ATP synthase, and this synthase is like a rotary motor. As the protons come in, they spin the ATP synthase. As the ATP synthase spins, this produces an ATP. Phosphate is added to ADP to make ATP. And because all of these redox reactions have occurred before we can make this ATP, this is called oxidative phosphorylation. That's a test question. The kind of phosphorylation that occurs in ETC is called oxidative because of all of these redox reactions that occur. Okay, so remember that it's called oxidative phosphorylation to produce the ATPs in ETC. It's called substrate phosphorylation to produce the ATPs in glycolysis and in Krebs cycle because no redox reactions are required to attach the phosphate to ADP. Now, this whole process here of spinning the ATP synthase and producing ATPs is called chemiosmosis. It's the manufacture of ATP via a proton gradient. The proton gradient means this. You have more protons that are positively charged on the outside of the Christie than you do on the inside, and so they're going to follow the law of diffusion and they're going to flow in. from high concentration to low concentration. Flowing in, spins the ATP synthase and forms an ATP. If you're a high energy bus, you're going to spin synthase 3 times. If you're a low energy bus, you only spin the ATP synthase 2 times. So that's why we say that high energy buses make 3 ATPs, low energy buses make 2 ATPs. Okay. So here's your summary diagram. What goes down the gradient? Hydrogen electrons. What gets pumped out from the inner compartment to the outer compartment? Hydrogen protons. Can those protons come back through anywhere on the gradient? No. They can only enter through ATP synthase. Now, the last electron acceptor is oxygen. These protons that come through the ATP synthase also join with oxygen and makes metabolic water. Then that metabolic water, remember, gets split up into hydrogen protons and the hydroxyl ions. The hydroxyl ions give us our negative charge. The protons that are pumped out give us our positive charge. And this flow of protons that generates the ATP is called chemiosmosis. Now, Peter Mitchell developed the steps of chemiosmosis and won the Nobel Prize for Chemistry in 1978. And all of those steps that occurred in Krebs cycle were elucidated by Sir Hans Krebs, and he won the Nobel Prize for Medicine and Physiology in 1953. So we have a summary chart of ATPs that are produced in ETC. Glycolysis, pre-Krebs, Krebs, Krebs. These are our sources of hydrogens. These are our types of buses. Glycolysis produces 2 low-energy NADH plus H buses. Pre-Krebs produces 2 high-energy NADH plus H buses. Krebs cycle produces 6 high-energy NADH plus H and 2 low-energy FADH plus H. So, this bus brings 4 electrons, hydrogen electrons, to ETC. These buses bring 4. These buses from Krebs bring 12 and 4. So, high-energy buses spin ATP synthase 3 times. Low-energy buses spin ATP synthase 2 times. So, if glycolysis has a low-energy bus, we've got 2 of them, it's going to spin it 2 times. So, glycolysis, the hydrogen from glycolysis, produces 4 ATPs. Pre-Krebs is a high-energy bus. So it's going to spin it 3 times per bus, making 6 ATPs. Krebs cycle has it spins it 3 times. They're all high energy. 6 buses gives us 18 ATPs. Krebs cycle for FAD, low energy buses, there's 2 of them, spins it 2 times, 4 ATPs, for a total of 32 ATPs that are produced in ETC. So if ETC makes 32 ATPs, Krebs made 2. Glycolysis made 4, net 2. You add that up, that's 38 ATPs, net 36. So eukaryotic cells make about 38 ATPs, net gain 36 from one molecule of glucose. Now, if you are a prokaryotic cell, a bacterial cell, bacteria don't have any mitochondria. And so all of these steps for ETC, instead of being on the cristae of a mitochondria, those proteins are embedded actually in the plasma membrane of the bacterial cell. So glycolysis, Krebs, ETC, all of that's occurring inside the mitochondrion. You don't have to pass through from the cytoplasm into the mitochondrion like the NAD buses from glycolysis in our cells. And so bacteria make 40 ATPs. Bacteria make 40 ATPs. Okay, that concludes our notes on cellular respiration, and I hope this helps you as you study for test 3.